Greetings, students, and welcome back to Chapter 9. Today we'll be covering sections 9.5 to 9.10. Where we left off in the end of the first section, we had looked at monohybrid crosses. And remember, monohybrid crosses are crosses between two individuals that are heterozygous for only one character. Today we're going to be looking at dihybrid crosses, which are crosses between two organisms that are heterozygous for two characters. So we're going to be actually looking at this relationship between two different characters and see what the chances are that you would end up with different combinations of those two characters. And this idea of looking at two different characters and tracking them helped lead to Mendel's law of independent assortment, which, if you remember from the last chapter, states that the alleles of a pair segregate independently of other allele pairs during gamete formation. So if we flash back here to the last chapter, remember we could have our chromosomes line up in this order, which will create pairs like this as we move through meiosis, which would then create daughter cells that look like this. But we could also have the chromosomes line up this way, which when they create them with a division that would look something like this. Notice now how different these daughter cells are here as we move on into the final parts of meiosis 2. Look at the difference now in the gametes. I could end up with character A and character B, or I could end up with character A here, but lowercase b here. I could end up with lowercase a and lowercase b here, and I can end up with lowercase a and capital B here. So they actually seem to travel differently. They're not necessarily attached to each other. It all depends on how the chromosomes assort themselves. And so this can be demonstrated using the dihybrid cross, which is this right here. The dihybrid cross represents all the different possible combinations of alleles when you look at two different characters. The traditional monohybrid cross is not big enough to represent all the possible combinations of gametes that exist when we're looking at two characters. Let's skip past these here and get to this one right here. Okay. So, and I'll go back to that in a second. This slide is kind of tucked in kind of weirdly in the chapter, but 9.6 mentions really briefly something we've already talked about in the chapter, and that's this idea if you have a particular parent and you don't know if it's homozygous or heterozygous, by looking at it you can't tell. The only way you could actually figure it out is by doing something called a test cross. It's where you mate that organism with a homozygous recessive individual, and their offspring will reveal what the actual chromosomal makeup is of the unknown. So if you look at this, for example, dog here, this could be a heterozygous black-furred dog or a homozygous black-furred dog. We don't know for sure. They both look exactly the same in terms of the phenotype. The only way you would know for sure is if you crossed them with the homozygous brown dog. And if you did that, you would end up with a chocolate dog in the offspring if this dog was heterozygous. If the dog is homozygous, you'll end up with all black because obviously the homozygous is dominant and would dominate all four part combinations. All right, so now back to our dihybrid crosses. Sorry for that little kind of side turn there. Our dihybrid crosses will allow us to calculate the chances of two traits appearing at the same time. For example, what are the chances of crossing two plants that are heterozygous for red flowers and being tall and getting offspring that are also red flowered and tall? So to do a dihybrid cross like this, we have to figure out first the parent genotypes, and we can figure that out by reading the question. Two plants that are heterozygous for red and being tall. Okay, so here's our key clue here, our context clue, heterozygous for red and being tall. Since they're heterozygous for red and tall, that tells us the red and tall are dominant. So I would write it like this, capital R, capital T, little t. Always put the alleles together for the same characters, like the R's with the R's, the T's with the T's. And always put the capital first and then the lowercase. That's the proper way to notate them. And it was this work, looking at two traits at one time, doing these dihybrid crosses, that helped Mendel to come up with this idea of law of independent assortment. So how does a dihybrid cross work? First thing we have to do is we have to take the parent genotypes and create the possible combinations of the gametes. Now remember before we had like a capital T, a little t, and that was it. We just divided them up. But there's more combinations than that in the dihybrid cross because we have to combine each character together in the two different possible orientations. So you can use the FOIL method to do that. You can take the first trait and cross it with the first trait of the other and combine it together. First the outer, then do the inner, and then the last. And that will represent all four possible combinations of gametes that can occur with those two characters. Now, you place those gametes across the top and the side just like we did with the monohybrid cross, and then cross them together, like this one intersects with this one intersects with this one, right? Put the alleles together, and then start counting them and looking for those phenotypes. Like red and tall would be anything with a capital R and a capital T, so you can count those up, and I would count 9 of them. Red and short would be anything with a capital R and anything with 2 lowercase t's, and so on and so forth. And when Mendel did his crosses of heterozygous parents, when he looked at 2 traits, it was interesting. He had a mathematical phenomenon, just like he did with the monohybrid crosses. But in this case, the traits always revealed themselves in the ratio of 9 to 3 to 3 to 1. So let's try one together. Here's an example of a cross between 2 heterozygous guinea pigs. I'm sorry, one heterozygous guinea pig, and the other one is white and short. Okay, so what I'm going to ask you to do is take this heterozygous black short-haired guinea pig and cross it with this white-haired, short-haired guinea pig. Okay, so the first thing you need to do is set up the gametes by using the FOIL method, then distribute the gametes to the sides, and then make the crosses. Okay, and then figure out how many phenotypes we have for each. Now be careful to note, for some reason here in this particular example, short hair is actually dominant, but it's the capital L. You would think it might be capital S, so please note that. So pause your screen for a second and give this a try. Once you're done, hit the play button and I'll reveal the answer on the next slide. So here's the answer here. Notice how I did this. I checked off each one of my phenotypes so I could keep track of them. I looked for all the black short ones, checked those off in red, looked for all the black long ones, checked those off in blue. I like to check them off so that way I make sure I keep track of which ones I've already included in my counts. That's a good technique to make sure you get these right. Another good technique is to make sure that the ratios always add up to 16. If you have more or less than 16, it means you did something wrong. Okay, if you have exactly 16, it doesn't mean you're 100% did it right, but there's a good chance you probably did. So those are the big mistakes people make. They miscount or they misread some of the phenotypes. Maybe you can't read your handwriting properly. So make sure they really discern from those capitals and those lower cases so you can see them clearly. Now, Mendel's laws actually reflect 2 different rules of probability. So if you take statistics, maybe in your junior or senior year, we'll learn a little bit more about this. We have the rule of multiplication, which calculates the probability of 2 independent events occurring. And then we have the rule of addition, which calculates the probability of an event that can occur in alternate ways. And we can kind of show this in the Punnett score here. So here we have our sex cells, okay? And we have one possible allele or the other possible allele, right? So we have a 1 out of 2 chance, okay? It's either one alternative or the other. But now what if I take that option here and I cross it with this one? What's the chance of these 2 actually intersecting here? That's where we get into the rule of multiplication. I have each one by itself independently is 1/2 chance. Bringing them together so that both of them would occur at one time would be 1/2 times 1/2. And so now we're talking about 1/4. Now here's something we're going to be looking at a little bit more towards the back end of the chapter as well. And that's this idea of a family pedigree. A pedigree very basically is simply a family tree, but it's a little more than that because it might also include information in terms of the genders of the people in the tree and also their genotype for particular types of traits. And so we can track traits passed through a family and study inheritance and find that many of the human traits that we look at are based on Mendelian genetics. For example, straight hairline versus a widow's peak. A widow's peak is a dominant trait. The straight hairline is a recessive version of this. So this particular trait, and this is part of the lab we did, does follow Mendelian genetics very nicely. Okay, and so there are examples. Did I skip a slide? Oh, here we go. Let me go back. I apologize for that. Here is a typical pedigree here. We can see based on the type of shape, it will indicate if you're male or female. Squares are male, circles are females. And then you'll see when it's highlighted in blue, that indicates the recessive version on this particular phenotype, if it's on this particular pedigree. If it's not shaded in, like here and here, that's the dominant version. So you can see in the first generation, both Al and Beth are male and female, and both have the widow's peak. Whereas on this side of the family, Charles has straight hairline, whereas wife has widow's peak. Move on into the offspring, and you can see here, you can follow the tracks. Since Charles is homozygous recessive and mom is heterozygous, it is possible to end up with a child that does have straight hair. Same thing here. Since both parents are heterozygous here, it is possible to pass that on to the offspring. And we call these parents here that have a particular genotype but don't exhibit the phenotype that you might find from that. And what do I mean by that? Let's say, for example, capital H, little h. In this case here, they both exhibit the widow's peak, but they carry the allele for the straight hairline. So they can actually pass it on. They're carriers. They will always exhibit the dominant phenotype, but they could express in future generations a genotype that includes a recessive allele. So their phenotype could actually change in the offspring. And you can see that, for example, in Frank and Gary. Frank and Gary both have straight hairlines, even though their parents both had widow's peaks, because they were both carriers. Now, some other examples of genetic disorders that follow Mendelian genetics, in addition to the ones that we looked at in the lab that we did together, are known to be inherited and controlled by dominant and recessive traits, just like we learned about with the pea plants. And like I said, those people who have recessive disorders but are born to normal parents, those parents are said to be carriers, and they're heterozygous, which means they carry the recessive allele even though they don't express it in the phenotype. So these are some different types of human disorders that are autosomal, which means they're in the chromosomes that are not the sex chromosomes. These are ones that are controlled by a recessive allele. These are ones that are controlled by a dominant allele. And then here's a couple examples. The freckles we looked at in the lab that we did, and albinism also follow Mendelian patterns that we learned about. And in the case of albinism, you can see here, could be expressed here, you have heterozygous parents. Both are carriers for the albinism, even though neither one of the parents exhibit, their skin tone is normal. But their offspring, there is a 25% chance that their offspring could exhibit albinism because they are both carriers. Now, there is different ways that scientists could ascertain genetic information. There's screenings that they can do, fetal testing, fetal imaging. There's different types of tests they can run, like amniocentesis, CBS test as well, which they can then give them information to use and to inform the parents. And this does create ethical dilemmas, because what if you do find out there's a particular disorder or some kind of malady or something that's discovered? That's a very could be very traumatic and stressful for the parents, and it presents a lot of challenges. And there's really not a lot that science can do today with some of these disorders other than identify them, but it does allow, for example, doctors and the parents to be prepared for when the child is born. So here's a couple of different examples of testing that can be done: the amniocentesis, the chorionic villus sampling, which is the CVS test, which will allow you to create things like a karyotype from the chromosomes, and could allow you, like we did in the lab that we looked at a couple weeks ago, look at the chromosome, see if there's an extra chromosome or a missing chromosome or a damaged chromosome, that kind of stuff. So that way that all the parties involved, like I said, from the doctors, the parents can be prepared. They can also do things like ultrasounds here or sonograms, and the doctor can capture this image of the baby here, and they could look for any kind of malformations or structural defects and things like that, which then the doctors can prepare for. Perhaps they'll need to know right away that some type of surgery is going to need to be done immediately after the baby is born, things like that. Okay. And so that's where we'll stop for today. And then for the last part of the unit, we'll look at some special cases that are non-Mendelian.